First of all, I would like to tell you about the features of DNA structure, some examples of DNA nanomaterial, some approaches for modeling of DNA structure, and also applications. And this is application in particular in lithium-ion batteries and DNA as a nanowire. So this is just some examples how it can be used. First of all, I would like to start with these famous papers by outstanding scientists James Watson, Francis Crick. You can see this first page, Maurice Wilkins, his colleagues, and Rosalind Franklin with Raymond Gosling. These three papers were published in one issue of Nature in 1953. And it was the beginning of new era in science, the beginning of molecular biology and many other very important and interesting research fields. What was known in that time? In that time, it was known that DNA consisted of nucleotides. One nucleotide consists of the phosphate group, desoxyribose, and nitrogen base, which can be one of the four types. This is adenine, guanine, thymine, and cytosine. And Watson and Crick, they understood that this nucleotide should form the pairs. Adenine always bind with thymine and guanine with cytosine. This is a famous rule, Chargaff rule. Erwin Chargaff, this is a chemist originated from Ukraine, from Chernivtsi. After that, he was born in Chernivtsi, and then he
I missed, was it? Я пропустила, хто це.
Erwin Chargaff. Erwin Chargaff, yes, he was born in Chernivtsi, and he was studied, he was graduated from university, and then he moved to the USA, and she worked in the USA. So, using this knowledge and also the experimental results by Maurice Wilkins and Rosalind Franklin, Watson and Crick understood how to compose model, DNA model, and they introduced this model in the shape of the double helix, and it was a great success. But this success was not possible without experimental results. And today we had a brilliant lecture by our guest, Professor Mats Larsson, about Nobel Prize. And here is also another one story. Probably you will find it interesting because the last one, the Nobel Prize winner, passed away just last year in October. It was James Watson. And after that, you can find maybe something interesting, but I'm sure that you will find, because this is almost detective story, how Watson and Crick found these experimental results. In fact, these results obtained by Rosalind Franklin were shown by Randall. This is the chief of Franklin and Maurice Wilkins. He showed these results to Watson without permission of Rosalind Franklin. And as was described in literature, Watson and Crick realized that due to this special spots in this image, that the structure of DNA should be in the shape of the double helix. And what is important that Rosalind Franklin was very careful experimenter. She did brilliant experiments with DNA. You can see these photos, the exposition time is about 100 hours. 100 hours. It's extremely difficult experiments. And she understood how to change the hydration of the samples. And in fact, she found the transition between A and B conformation of the double helix. So if we decrease the hydration, as a double helix in the shape of A conformation, and if we increase hydration, this conformation changes to P4. And so I will continue without my slides, but in general it is possible to describe this because there were it was another one paper published in Arte Crystallography Journal, where Rosalind Franklin also introduced such scheme of neutralization of the phosphate groups. Here you can see each phosphate group is negatively charged, and to make neutral the system, she suggested that ions should be added to the phosphate groups and neutralized in such way. So we will return back to this scheme due to the other problem. And here I just want to show you what I told before, that the story about discovery of DNA, about the story about Rosalind Franklin was in performance photo number 51. This is the photo that I showed you above. And the main role was played by Nicole Kidman. And this year, the movie about the story will be produced with the main role with Natalie Portman as Rosalind Franklin. So, and about unique properties, I already told that nucleotides form the hydrogen bonded pairs, and it makes possible the construction of very interesting materials. Like you can see, like boxes, we can have different shapes of this material due to the self-assembling properties of DNA. And the next one property which makes DNA unique material, as unique material, you can see that the phosphate groups are in every nucleotide, and then as a result, DNA is highly charged. And in the solution, these charges are neutralized by different ions. We call this counter ions because they neutralize the opposite charge. And usually this is metals. It may be some metals like sodium, potassium, but also other different metals which we can add to solution. And the last one feature of DNA that I would like to highlight is that DNA is common in the world. And it is very interesting that there is estimation of the amount of DNA. And it is, if we take this in kilograms, so this is something about 5 multiplied by 10 in power 13. And this amount is comparable to the amount of oil on the Earth. So the amount of DNA is huge. And that's why this material can be used from in practical sense. So the interaction of ions with DNA is very important, and here I will be focused in use of this property of DNA with the ions. And here you can see this scheme which we introduced with our colleagues Dmytro Pyatnitsky, Tatiana Buban, and our colleagues from abroad, Narcis Shibotario, Arthur Laksonen, and Francesca Mucci. We prepared this review where we highlighted these properties of DNA with ions, which can be used in different applications. And today I will tell you in brief about some applications of DNA in batteries and also as metallized DNA. As metallized DNA, so the ions which are embedded inside the double helix. To understand the interaction of DNA of ions with DNA, we need some approaches, we need some models, and the simplest model is the model where DNA is considered as a chain of charged beads. You can see this very simple model, but at the same time, it allows us to to find new very important effect, which is known as counter-ion condensation. And within the framework of this model, it was shown that the ions form a cloud around DNA, and this result was supported by many experiments. Of course, there are also other more complicated models on the basis of Poisson-Boltzmann equation, which we cannot solve analytically, but in some approximations, like the bifugal approximation, we know the solution, and also other approximations like the Kachalsky approximation, and many, not so many, but still we have some approximations allowing us to obtain analytical solution. What is interesting is that this problem can be reduced to the Penrose-Lee equation, which can be also studied, and this may be very interesting application of this equation to this particular problem. In our institute, we introduced another approach where we present DNA with ions as a lattice, where each phosphate group is neutralized by positively charged ions, ions. It is different. This approach is different because in the case of all these models, simple polyelectrolyte model and Poisson-Boltzmann equation models, in these models, ions are presented as a uniformly charged continuum. But here in this model, in our approach, we present the system as a lattice. And we obtain also very interesting analytical results, which we can compare with the experimental data. But of course, this is not enough to explain a very complex system of DNA with ions, with water molecules, which are everywhere around. And in this case, we have to face to numerical methods, to simulations, and it may be done on different levels. First of all, this is the basic level is electronic level, where we can use well-developed quantum chemical approaches. Then we can move to atomic level, where each atom is presented as a sphere, and the spheres can bind to each other, and we can describe this interaction using interaction potentials. And this approach is also well developed and can be used for rather large systems. And if we want to describe a larger system, we need to build coarse-grained models. So we have to grow the atoms to separate masses, and we have to describe the interaction between this growth of masses. So this is coarse-grained models, which allows us to model larger DNA molecules in larger time scale. In general, we can highlight this way of description as an electronic system, as a real system. After that, we have a sketch of this real system figure, atomic level, and then coarse-grained level. This is like this picture where you can still recognize this bird, but in fact, it looks very rough. Next, I would like to describe some results which we obtained within the framework of atomistic molecular dynamics simulations. And these results, we have a lot of results. I mean, in this field, in the field of the study of interaction of ions with DNA, there are a lot of papers about this problem, and we can start discuss these results from 80s of the past century. But I would like to stop on some results that we obtained here at the institute.
Metal or metal? Effects of hydration.
Of metal, of course. Sorry, there is missing letter. Thank you very much. Yes, you.
Well, also milk, why not?
Yes, yes, yes. And yeah, this is the metal. You can see metal, and this is a hydrated metal. They are hydrated. You can see this some ions without hydration shell because they are negatively hydrated, and some ions are positively hydrated, and they constrain water molecules in the hydration shell. So, like magnesium ion, for instance, you can see it is highly hydrated. And the cesium ion is not is negatively hydrated, and it can bind to DNA without hydration. And this system is very interesting because, for instance, in the external electric field, the ions move, of course, and our molecular dynamics simulation shows that this ion, which are close to the DNA double helix, they follow the structure of DNA. So the ions move along the grooves of the double helix. And comparing these results with the experiment, this experiment appear later than we have done the simulations. We can see that the diffusion coefficients of the ions which are close to DNA, and in a free solution, the ratio of these two diffusion coefficients is almost constant. So it is about 0.6. And we compare our results that we obtained for potassium ion, and we found that this result is completely in trend with this experimental data. And if the field will be perpendicular to the double helix, we will have melting of the double helix. We can have observe such called field-induced melting. DNA breaks, the hydrogen bonds break, and also we found in literature that there were simulations where DNA melts under the influence of alternative electric field, and it was shown that it depends on frequency of this field, and this process is facilitated. Another one very interesting simulation was done for DNA in solution with ions, and it was shown that the external electric field made DNA macromolecule rotating. And it was shown that this is because ions move and they induce the water flow, making this rotation possible. So in such a way, the authors have an idea of such tiny electric motor, and this is possible, and but this is haven't done yet experimentally. This is just atomistic simulations. But what we can say about real experiment and real application? Real application is very interesting in the case if we are talking about DNA batteries. Here you can see these figures from publications of scientists from South Korea, where they constructed a supercapacitor. Here you can see this. This is a sketch, of course. The electrodes and the DNA gel between them. So this DNA gel looks like in this picture, and in the scheme, this gel can be presented like this, like winded DNA macromolecules with some lithium ions between them. But in fact, we don't know how this gel is organized. And this is one of the questions that should be answered, because in fact, this scheme is very rough, and we need to specify. I'm very glad that we have collaboration with our colleagues from Taras Shevchenko University from physics department, where we have studied the conductivity of DNA solution with lithium chloride and compared this conductivity without DNA, conductivity of lithium chloride without DNA. This is actually a rather simple problem. I mean, simple idea, but in fact, we obtained a rather interesting result, which show us that the difference between conductivity of DNA with lithium and just lithium without DNA is not monotonic. So we have some decrease of conductivity with increasing of concentration, decrease of difference of conductivity. After that, we have some plateau, and then decrease again this difference. These different lines correspond to different temperatures and as well as in these figures. And we explain this result as trapping of the ions. So DNA trap the ions, but it trapped too much. We need to explain this result. We need to trap up to 100 of ion pairs in some region. And of course, it is I mean 100 ion pair per one phosphate group. And of course, it means that we should expect some regions where this ion is trapped, and these regions we suggest is the region with low dielectric constant. And in these regions, the ions are constrained. And in this case, we can explain, we can obtain such behavior of electrical conductivity. But of course, such simple model cannot explain all peculiarities of the system, and we need to perform molecular dynamic simulations. And here you can see the results that we obtain using our computational cluster, where you have 5 DNA molecules in the simulation box with water and ions. You can see just only ions. Water is not shown here. The ions are positively charged. This is lithium, and the negatively charged chloride. And we wanted to find what will happen with the system, and we found very interesting effect, which was actually a big surprise for us, because we found that these molecules stick to each other. And you can see here how it looks like. There are different molecules. They form the aggregate, and everywhere we have lithium ions. So this is a surprising result, because we have it is known that to induce the aggregation between 2 different molecules, we need to add some charges, some counter ions with high charge, something about 3+ and greater. We have monovalent ions, and we didn't expect that we should have such effect of aggregation. And we wanted to understand why it happens. So we analyzed the structure, and we found that in the case of lithium, we have overcharging of DNA. What does it mean? There are some phosphate groups where we have 2 lithium ions. You can see these red circles showing us these phosphates with 2 lithium ions. Also, of course, we have the phosphates with squares with 1 lithium ion, and some phosphates without ions at all. So what we have? We have the system where the phosphate groups are neutralized very intensively. And in fact, I just would like to say that this system of DNA with lithium ions is the closest appear as the closest to our idea, our concept of the DNA iron phosphate lattice, where we presented DNA designs as ionic lattice. And when we have overcharging of DNA, we can see that different regions of DNA where we have positive charge due to two lithium ions and negative charge regions, they can come together, and in such a way we have aggregation. So actually this mechanism is common, and it happens if we consider the system with polyvalent ions. But in the case of monovalent ions, the effect of overcharging was not observed. And we observe here in the case of lithium ions. So lithium neutralizes two oxygen atoms of one phosphate, and another one phosphate without lithium comes to the contact, and in such a way we obtain the cross-link between two different phosphate groups. We took several complexes of such kind and from electrodynamic simulations, and calculated using quantum chemical methods. It was done by Dr. Metallized, and we found that such complexes are very stable. And in fact, what we found is that this is like crosslinks which were introduced by Rosalind Franklin, but instead of sodium, we have lithium. And why lithium was here is that because lithium is highly hydrated. This ion is very small, and the hydration flow of the ion is very strong. And as a result, you can see here these two water molecules around lithium, and also another two oxygen atoms, but not from water molecule, but from phosphate group. And this structure is very stable because this is tetrahedral structure, like a structure of lithium ion surrounded by water molecules. Another important feature that I would like to tell is also related to conductivity, but not ionic conductivity, but electrical conductivity with electrons, proper conductivity of DNA. So this question arises probably from the very beginning. Yes, I'm finishing. From the very beginning of study of physical properties of DNA, and here you can see the scheme of experiment where you have two electrodes, and between them you have DNA, and you can measure different volt-ampere characteristics at different temperatures and so on. Actually, proper conductivity of DNA is more insulated. And if we are talking about the mechanisms that may be in this system, the conductivity mechanisms, this is like a tunneling of electrons, or hopping from one nucleotide base to another. And to increase the efficiency of such a system as a conductor, we can face to metallization of DNA. So if we put inside DNA macromolecule metal ions, for instance, argentum, you can see here, this is the experimental results, experimental data, X-ray experimental data, and inside of DNA you have argentum ions, and they form such a chain that may be considered as a wire. Also, this is the result by Japanese growth, and also there are other results. This is X-ray, this is results in NMR obtained by growth from Slovenia, from Ljubljana. You can see here the same silver atoms are inside DNA, and they have such slightly different organization inside DNA. And a little bit other story about metallization is that it is possible to expect that metal clusters can be embedded inside the DNA, intercalated inside DNA, and this is also very interesting story for the. So to understand the stability of the system, we have calculated the complexes of argentum ions with different nucleotides, thymine, guanine, adenine, cytosine, in different positions. We used DFT method, different functionals, different basis sets. And what is interesting, we found that the most stable is the complex of argentum with guanine, and the adenine is the least stable. And when we compare these results with experiments, we found that adenine is pushed out from the system. You can see these nucleotides, they are not inside the double helix, they are outside. In this scheme, you can see there are 4 such nucleotides, and this is adenine. So adenine is the least stable, and that's why it is pushed out by argentum, and in such way the system is formed. So the following step for us is to understand how this chain is formed, and we need to calculate the interaction between different argentum atoms to understand how the argentophilic bond is formed. So in conclusion, I'm finishing. I would like to say that DNA is a very promising material. So in fact, it is the material and can be used in different applications, in particular due to conducting properties in ionic conductance, and also due to metallized properties, due to the properties of metallized DNA. And I think it will be developed in the nearest future. So in the very end, I would like to thank my colleagues from the Bogoliubov Institute for Theoretical Physics, Professor Volkov, Dr. Grechko, Dr. Pятницкий, Dr. Tatiana Bulban, Dr. Zdarevsky, Dr. Osokin, Bogdan Reshovka, Polina Konevska, Dr. Vitaly Tomchishin, my colleagues from different countries. I will not mention them, but it is this collaboration is very important. We did a lot of very interesting research, and also I'm happy that we have now collaboration with our experimentalists from Parashevchenko National University, headed by Professor Bulavin, and also Dr. Lazarenko, Dr. Katerina Yavlichkova, and others. I would like to thank you for your attention.
Question. Thank you very much for nice and promising report. You must look to general time. Is it possible maybe in future to use such structures by DNA for writing and reading information, because it is a natural store of the information?
Thank you very much. This is a very interesting question, and you can find even in Wikipedia DNA computer. There is such term, and it already exists. So the writing the information works on the basis of chemical reactions. It is very slow. Actually, in this case, I often show such video where you can where some computer is demonstrated as people who show different flags and so on. So it is possible. So also it is possible to construct, and it is already built, the computer, the memory on the basis of DNA with reactions, it is very slow, and but nevertheless people consider that it is possible to use such computer if you don't care about time of writing. So if you need to save some information for the long time, you do this and it will be saved. And the process of writing can be long, but it is not so important. So it already works. And another one feature that I would like to tell about this is that what I found in some recent paper, scientific reports to 2024, that the title of the paper is DNA as a promising material for quantum computer. So, but this is more speculation. But still, people think about this, and I think that it is really, really possible.
Thank you. Is it on? Okay. Thank you for very interesting talk. I was intrigued by your comparison between the amount of DNA and oil. Where is all this DNA stored? Is it in human?
You mean in the Earth?
Yes.
Yes, this is the paper in PLOS ONE in 2015, it seems to me. I was surprised when I found such analysis, and what they have done, they just used open information about DNA, and what they about 2015 open information, and they just estimated how many DNA we can find in bacteria, in viruses, in different organisms. So this information is available, and they had a big table with all this data, and they estimate. Oh, and about oil, this information is in Wikipedia. So
Yes.
I just took it.
May I? Is there any question in Zoom? Meanwhile, I will ask, first of all, thank you very much for your very interesting talk. One is very boring question. You have considered high temperatures with lithium, like 65, 50 degrees, but the DNA melts at these temperatures. And other two questions, brief but more qualitative. So about
about this, you are completely right. If we increase the temperature up to 65, we have melting of DNA. And I wanted to take this into account in my model, but we had so many parameters that I give up. No, not give up. I mean, we need to give the answer. Restrict a little bit. But this problem is not so clear because you see we have also very large concentration of ions, and increase of concentration increases the temperature. So the temperature of melting temperature may be even higher, even about 90 degrees, something like this, because the concentration of ions is huge.
That was my question. Another one. Yes, thank you. And another one is, we know that there is very strong pollution with the technology's advancement of metal pollution. Do you think that your research, your results can be related to the impact of the metal pollution on DNA metabolism of the living systems and probably also cancer?
Actually, I return back to this review that should be published just.
We are waiting.
Yes. I advertise our results. So we discussed this, and of course, DNA is considered as some material that can be used to take these heavy metals, for instance, or as in some medical applications and many others. So that is true. That is true, and DNA is have a lot of different applications. I just discussed this one and this one, and probably just mentioned about origami, but also we have many other applications of DNA and the iron. So this combination, I mean, this property of DNA that it is highly charged polyelectrolyte, it makes very important. This is very important property as a material.
So the last question from these two. Oh, you really start.
I'm sorry if I missed it in your talk. Do magnetic fields play any role in the dynamics of the DNA, either external or internal, either due to currents, either due to some multiples, molecules, and our spin?
Thank you. This is also a very important physical question because, in fact, for DNA, not. But if we are talking about metallized DNA, it is very important. It is very important that I even discussed this question with Volodya Kravchuk. We had such interesting conversation about this because this ion may have some spins and so on, and in this case, magnetic field will play a crucial role.
My question is are there some analogous properties, effects possible for triple helix structure?
Of course. We have our Doctor of Honoris Causa, Professor Maxim Frank Kamenetsky, and he is one of the co-author of the discovery of triple DNA. So this object exists. Yes, that's happened. So it is possible, it exists, and it even has some biological meaning, importance.
So discoveries are there.
Yes, it works. Yes, of course, it works. So triple helix, we also have quadruplexes. Quadruplexes are also possible, and they are very important in biology, in telomeres. In our institute, Professor Perechko works with quadruplexes. So this is.
I see some paper about epimophysics.
Ephemeral physics, I remember Ivan Vasilyevich Simonov about this, and this is really interesting. Unfortunately, I just in brief discussed with him this, and I had no time to go with this problem. So, yes, this is very interesting. Let's start.
Otherwise, if we consider dynamic continuum, you sure it's actually not dynamic, is it? And it's well known that in any discrete quasi-one-dimensional system, there are at least two possible effects. One effect deals with the so-called Poincare number problem when the size of transmitting particle is comparable with the distance between the elements of structure. Yeah. It's possible to pinning these transmitting particles or ions or maybe in such structure. And another well-known effect is so-called Bloch oscillation, when the electric field or any field as applied along the chain. It's possible also the so-called Bloch oscillations. So the Bloch oscillations are caused by the periodic form of dispersion law. We can
say about these two effects in your consideration?
It is, these effects are very interesting, and I had an idea just somehow to study some nonlinear effects using molecular dynamics. But in fact, this is really a complicated problem. And of course, we can build different models, analytical models, as you described for DNA, and they are built. And for instance, the models which are developed by Professor Sergiy Volkov, he did a lot in this field. But it is very difficult to detect them, and the idea is to study somehow within the framework of simulations, molecular dynamics simulations, for instance. But even in this case, this is not easy task. So this is probably just open questions, because there is a gap between some results that you describe in analytical models and experiment and simulations. So computational experiment and real experiment. So this is in the case of DNA. I don't know, maybe for some other systems you can find, but for DNA it's not so easy.
Thank you, sir. Thank you. So, exactly. So no. So,
Okay.
we have 10 minutes.